Delighted that you're joining us this evening from across Australia, and I'm really excited to share with you this webinar from Professor Caroline Mansfield called All Guts and Glory. She's going to shed some great insights into the microbiome for us. There's a huge area of focus of research at the moment globally in the veterinary and the human field. Before I introduce Caroline properly, just a quick housekeeping note. We are recording this session, so it will be available on demand afterwards. And if you have opted into receiving communications from us, we'll be able to send you a link to that on-demand viewing. We're also hoping to have time for questions at the end of the recording and at the end of the session. So if you can hold your questions, pop them into the chat function as Caroline gives her talk, then we'll be able to get to them at the end. We'll leave some time, about 10 minutes at the end for those questions. All right, well, without further ado, it's my great honour and privilege to welcome Professor Caroline Mansfield joining us tonight from here in Australia as well. She graduated from Murdoch University in Perth as a vet in the last century, and that's according to her own words, not mine, although it makes me feel old too. She's worked in mixed and small animal practice in Australia and the UK before she completed a 3-year residency in small animal medicine at my old alma mater, University College Dublin in Ireland. Professor Mansfield developed her interest in gastroenterology during that time back in Ireland, and she's continued that clinical and research passion since she returned to Australia. From 2001 to 2010, she was employed at Murdoch University as a clinical registrar and then lecturer, followed by senior lecturer. She then moved to the University of Melbourne in late 2010 as the head of small animal medicine, and later became professor and director of clinical research, finishing there in mid-2021 or mid-pandemic. She currently holds a research position at the University of Queensland. So her interest has been focused on the mechanisms predominantly involved in canine inflammatory bowel disease, the endocrine and exocrine pancreas, and establishing the impact that the gut microbiome has on metabolic health and disease in dogs. She is board certified in companion animal internal medicine and gained a diploma at the European College of Veterinary Internal Medicine in 2001. She is past president of the Australian and New Zealand College of Veterinary Scientists and was a board member for Vet Compass Australia, which used big data to track breed-related health and disease. She enjoys clinical case management, clinically relevant research, mentoring and teaching, and unbelievably also has a life outside of all of this with a family where she has 3 teenage children, 3 cats and 1 new puppy. So Caroline, I shall hand over to you now. And thank you so much again.
Thank you very much for that wonderful talk. Hopefully everyone can see my slides now.
Yeah, perfect.
Terrific. So I'm really delighted to be asked to speak to you tonight about an introduction, I guess, into unraveling the beauty that is the GI microbiome and the impact that it has on systemic health and potentially manipulation and management of a multitude of health problems. Before we start, I have the following relationships to disclose, although I don't think any of them will directly impact this talk. So what I'm going to address in my talk briefly for each of these aspects tonight, I'm going to start off just by defining what the microbiome is, how we can assess the microbiome, and there's a difference, I guess, between how we assess it clinically and how we do it in research projects. What we know about how the microbiome impacts health and disease and how it interacts with various conditions. And then I'll just spend a little bit of time talking about the options and the ways in which we can manipulate the microbiome at the end. And hopefully it will stimulate some conversations and questions at the end as well. So just to recap, the GI microbiome is the collection of microorganisms that lives within the gastrointestinal tract. And mammals contain about 10 to the 12th to 10 to the 14th bacterial organisms, which means that there are as many bacterial, DNA weight as there are human DNA. There are over 500 bacterial species in dogs and over 1,000 in cats that have been detected. And there are 5 major intestinal phyla, so Firmicutes, Fusobacteria, Bacteroidetes, Proteobacteria, and Actinobacteria. It's important to remember that the microbiome changes along the gastrointestinal tract. So when a lot of papers and a lot of studies refer to the GI microbiome, and it's probably the fecal microbiome, and there's a slightly different distribution in terms of what bacteria are present, with Fusobacterium, Bacteroides and Firmicutes being the most prominent phyla and Clostridium being the most prominent genus. There are also lots of other things that that comprise the GI microbiome other than bacteria, although these are less studied, probably because of the techniques and methodologies that we have. So we know that there are viruses that outnumber bacteria, but only contribute about 2 to 5% of the total DNA of the GI microbiome. And these viruses are maybe more important than we anticipate, and we might be learning a lot more about them in future, particularly bacteriophages, which are virus-like particles that can actually drive and manipulate the GI microbiome. And then there are also archaea and fungi, which are relevant because they produce methane and other products. So there's been a huge explosion in knowledge about the microbiome in terms of the number of publications with, you know, even as far away as 2017, there are over sort of 10,000 articles produced on the microbiome, looking at things such as soil and water microbiome, and about 3,000 publications per year on the GI microbiome. And the reason why there's been this explosion in knowledge is because there has been a development and more accessible method of detecting and identifying these bacteria that doesn't rely on culture, which used to be the traditional way that we could assess and what bacteria were present. And we know that a vast majority of bacteria that are present within the GI tract actually can't be cultured. So the non-culture methodologies that have probably been most prominent in research and even in clinical aspects of GI medicine include fluorescent in situ hybridization of fish, quantitative real-time PCR. I'm not really going to talk about qPCR that much, but qPCR is when you have an identified and specific bacteria or pathogen, and you're able to have probes against that particular genetic sequence for that organism, and you're able to estimate the quantity as well as the presence of that particular bacteria. And then there's also next generation sequencing, which I'll just talk about a little bit more as well. But when we talk about fish or the in situ hybridization, this is when we actually have probes against bacteria that are actually applied to tissue, and so that we can see spatially where these probes or where these bacteria are present, and we're able to have specific probes against sort of particular bacterial genus or sometimes even species. So the veterinary application in which fish is probably most utilised and most known is trying to identify bacteria in particular forms of GI disease such as granulomatous colitis, which typically affects French bulldogs and boxers. So this was from the very first paper that was sort of outlining that adherent and invasive E. coli was associated with granulomatous colitis. And the picture on the left, labelled A, is a normal intestine, you know, colonic crypts, and that you can see the red bacteria are just sitting in the mucus, not invading into the tissue. Whereas the bacteria on the right, which is a pretty inflamed and ugly looking colon from a boxer, you can see the positive red staining throughout and deep within the tissue, and sort of surrounded as well by inflammatory cells. So when we talk about next generation sequencing, or the term that's often called 16S sequencing or pyrosequencing, this is where a lot of the studies in human and veterinary gastroenterology have started. And so this is where in a particular sample, whether it's an environmental or a biological sample, the bacteria are amplified using a universal primer. And so the primer that's most commonly used is 16S ribosomal RNA gene. And this gene is universal to bacteria. Once these sequences are out there, they are then sequenced, and you have a huge number of potential sequences of various quality and various read lengths. So there are a whole bunch of bioinformatic platforms and methodologies that are used to trim those sequences for quality and then the sequences that are left are matched to known databases, of which the most known or best known is a platform called CHIME. So next generation sequencing or 16S sequencing can tell us the bacteria that are present probably to a family level and rather than a species level. There are a lot of false positives and false negatives that you can get with next generation sequencing, and that analytical platform or the analytical pathway can be quite variable between different studies. And so sometimes it is difficult to compare one study's finding to another because of the differences in the way that the study has been conducted. The other drawback with 16S sequencing is it doesn't actually tell you what the function, or what the impact of those bacteria are. And we know that within the GI microbiome, there's a lot of redundancy of roles. So if one bacteria is depleted, there may be another bacteria that is able to overtake that particular function. We also know that there's a codependency between some particular bacteria as well. So in order to overcome that, there are some predictive models that can be used that can try and predict whether there's changes in the functionality of the bacteria based on the expected genetic function or genetic coding of the bacteria, but it is an extrapolation or a hypothesis or a guess. Although this seems to be predominantly a research tool, in a lot of countries now, sequencing of the GI microbiome using this methodology is available to members of the public and their pets to send through. So it's something that I think will eventually become more common in Australia as well. What I will say as well, though, is that particularly with GI disease, the GI microbiome is highly labile and highly variable from day to day, and even within a day, and it's highly individual as well. So a single one-off sequencing to me doesn't really help and shouldn't drive decision making in the clinic. Certainly sequential sequencing is maybe a little bit more relevant. So something that is becoming more frequently done is called metagenomics or whole genome sequencing, and that's when the genomic DNA is fragmented. Now, we can't do this on tissue because of the amount of host DNA, but we can do this on feces and then filter out any mammalian sequencing that is identified. This actually allows us to identify bacteria down to a species level, but also their encoded genes. So it's better at capturing the functionality of a bacterial community. The reason why this hasn't been done as extensively as 16S sequencing is that, prior to the last 3 to 4 years, it was incredibly expensive. So it was, $400 or $500 per sample. You do end up with a lot of data. You end up with sort of 24 million sequences that you do have to assess per sample, but it does allow a greater level of detail to be known. So the other thing, I guess, that we can that is also becoming more apparent in a lot of clinical studies is a term called metabolomics. And so even though we know what the genetic coding for bacteria are using, if we do whole genome sequencing, or we can guess what it's going to be using 16S, it doesn't actually tell us whether the bacteria are changing their function. So with metabolomics, you can assess various bacterial metabolites within the feces, but also within the host. And it can be targeted or untargeted. My preference is that that studies use targeted metabolomics, so assess what the bacteria are, and then target the change using different mass spectrometry methods. And there are also some centres where fecal bile acids, which I'll talk about a bit later about how important they are, they can be measured independently or sort of standard metabolomic analysis as well. And something that is probably more frequently used, both clinically, but also in research methodologies, is something called a dysbiosis index. So the Canine Dysbiosis Index was published in 2017, and with this there are 9 quantitative PCRs, so a universal primer or the entire bacterial load, and then using a comparison between healthy dogs and dogs with chronic enteropathy or chronic diarrhea, they identified 8 other, they identified, I can't add up, they identified 7 specific bacteria that were different between those 2 groups. And then they came up with a dysbiosis index as a result. So using a mathematical model and looking at the expression of each of these bacteria and the universal primer. And with this dysbiosis index, a negative dysbiosis index or a negative DI, usually less than -2, means that the microbiome is considered normal, whereas a positive dysbiosis index correlates to a dysbiosis. And so when we look at these, the C. hieronymus or Clostridium hieronymus is one that is commonly depleted in dogs with chronic diarrhea or chronic enteropathy. And this is a really important bacteria because it's uniquely associated with bile acid metabolism. And we know that bile acids can be altered both with chronic and acute diarrhea. So there is also a dysbiosis index that has been developed in cats, and this was more recently published, so it was just published last year. You know, as with all cats, the overlap between healthy and cats with chronic enteropathy is much greater. And we find as well that if the cats are anorectic, rather than having vomiting or diarrhea as their predominant clinical sign, they are more likely to have a dysbiosis using the dysbiosis index. Now, both of these are actually available through the Texas GI lab, and so it is feasible that samples can be sent to you know, to the US to have this measured. The biggest issue at the moment is shipping and transportation. So why are we interested so much in looking at the microbiome? So we know that in people there is a rapid change in the way that the microbiome develops, and there's a lot of suggestion that the GI microbiome is established at an early age, and it's very much altered by diet in infancy, and then it's affected by antibiotic treatment and nutrition as children are growing. And then we also find that there is a different phenotype between healthy and obese adults. And there's also a different phenotype in the GI microbiome between elderly patients, but then also those that are considered frail. And so, as well as a difference in the actual distribution of bacteria, or what we call the diversity, there's also a decrease in richness as people get older. And there's probably a similar thing in dogs. So this is from a study that we did in guide dogs. So we looked at guide dogs from birth up until 12 weeks of age or 40, and we took weekly fecal samples, and then we compared these to Labradors that were middle-aged, so between 2 and 7, and then elderly, so Labradors that were over 10. And so we did find that there was a decrease in richness and an alteration in diversity as the dogs got older, and there was a vastly different microbiome in puppies. And the diagram on the right shows that this shift in the microbiome is incredibly rapid, and it occurs right around the time of weaning, so around week 4 to 5. And then these are the times of which there is the most rapid change. So during this time is when the GI microbiome is developing in puppies, when it's changing rapidly, and it's also interestingly where the biggest neural development and behavioural development of puppies is occurring. So what does that mean, or what impact could that have? Well, there's now been 2 studies that have shown an increased incidence of health problems in later life, particularly GI diseases in puppies with parvovirus. And this hasn't been shown in dogs with acute hemorrhagic diarrhea syndrome. And the reason why this might be different is because, obviously there's the developing microbiome in these puppies, and also the parvovirus wipes out the villi and the normal GI sort of structure and format. more so than AHDS is. But probably the other reason is that most parvo puppies are actually given antimicrobials because they're septic. And so it's thought that these really early childhood antimicrobials might have a really long-term impact on the microbiome and the metabolome as well. What hasn't been studied yet, and I think would be very interesting to study, is whether puppies with parvovirus and treated with antimicrobials have a higher incidence of behavioural abnormalities when they get older as well. And you know, I'm just bookmarking that as a study that I'm really interested in undertaking. So what we do know is that the diet is probably the biggest influence in dogs and cats, and it's probably all components of the diet, so not just protein, but also fat, carbohydrate, and fibre that will all contribute, and it's the quality of each of those subcomponents, not just the quantity. So there was one study that looked at dogs fed barf, so a raw food diet compared to a commercial food diet, and there was a significant difference in that dysbiosis index. Now, I want to be really careful and say that dogs that are fed a barf diet, they don't necessarily have a dysbiosis. In this study, they were all considered to be very healthy, but there is a difference in this quantitative PCR index between those two different food groups. So what is the role of the microbes in health? So they ferment carbohydrates, which is really important. And probably more important is the production of short-chain fatty acids, in particular butyrate and acetate, that are important for reducing systemic inflammation, but also improving colonic health and also as an energy source for the host. They're also very important in synthesis of a variety of vitamins. They are also responsible for a whole bunch of neuroendocrine functions, such as making sure that there's not too much tryptophan. And they also deconjugate primary bile acids to secondary bile acids. And this is, again, an emerging area of interest because bile acids have been shown to be really vital in regulating the amount of fat that's absorbed from the intestine, regulation of insulin production, and also have direct anti-inflammatory functions. And we know that with some chronic recurring GI infections, particularly Clostridium difficile in people, there is a massive alteration in the bile acid ratio, and a Western diet predisposes to increased secondary bile acids in the colon, and that's thought to be one of the reasons why there's an increase in intestinal cancer as well. So why are we interested clinically in the microbiome? What applications or what thoughts might we be thinking about in the veterinary field, and why is it so important? You know, I've talked a little bit about what its role is in health, but I think what's probably a nice way to think about the gut is it's actually probably the biggest immune organ in the body, and it's the major interface between the host's immune system and the external environment. And in dogs in particular, the exposure to foreign antigens is virtually constant. There is an innate and an adaptive immune system within the GI system, and the gut, we know, does interact with distant organs as well, which I'll talk about in a minute. And it's really important for this mucosal immune system to work effectively to have bacteria present. And there's a whole bunch of studies that have shown that when animals are born germ-free, so when they actually have no bacteria and they're not introduced to any bacteria, that there are a whole bunch of functional, immunological and anatomical changes to the GI system, and also an increased susceptibility to systemic infections and autoimmune disease as well. So having a good microbiome is incredibly important for health. And we also know that there is a dysbiosis that occurs in inflammatory systemic disease. Now, in people, there has been a really marked association between dysbiosis and metabolic diseases like asthma, HP, diabetes, and obesity. And there's a landmark Canadian study that looked at the incidence of asthma in children. And so it used to be thought that for example, being formula fed or cesarean born increased the risk of HP. But what they've actually found is it's actually these 4 gut bacteria being present in the first 3 months of life. So Faecalibacterium, Lachnospira, Veillonella and Rothia, and I probably mispronounced those. And irrespective of anything else, so environment, diet, method of birth. And so if you have these 4 gut bacteria, you'll have a decreased risk of asthma. And this systemic inflammation and the systemic impacts of the GI microbiome is actually amplified in the face of obesity. So we know that when there's obesity, that there is a leaky gut, and there's more likely to, as a result, to be circulating low levels of endotoxemia, so bacterial LPS, and changes within the liver as well, including insulin resistance and oxidative stress. So all of those systemic inflammatory effects are amplified with obesity. Additionally, there's an increased incidence in the obesity phenotype and how potentially the microbiome can impact and cause or, amplify obesity. And I apologize for these graphics. They are very, very poor graphics. But in the initial landmark study that was performed in 2008, so there were 4 colonies of lean mice. 2 of those colonies were transplanted with an obese microbiome, so obese mice, and then 2 groups were with a lean microbiome. And out of those, 1 group from each microbiome was fed either a high-fat Western-type diet or a more of a healthy, low-fat chow. And what they found was that the mice that were transplanted with the obese microbiome became overweight regardless of what diet they were fed. And similarly, those that had the lean microbiome could eat as much of the high-fat diet as they could, and they remained lean. So then this was sort of further expanded by the same group, and they actually cultured gut bacteria from identical human twins that were discordant for obesity, so 1 overweight and 1 normal. And what they actually found was that there was a difference in the microbiome between those two groups, and that when they actually transplanted the obese human microbiota into germ-free mice, the ones that were transplanted the obese microbiome became obese as well, and that was irrespective or regardless of the diet that they were fed. And so this then, when they were co-housed, so when the obese mice were able to eat the lean mice feces, so kind of like a self-induced transplantation, no obesity actually developed. And so this rescue was associated with a Bacteroidetes, and it was enhanced by diet, but it was associated predominantly with the other interesting thing is that when we're talking about weight loss in people, that the most successful weight loss treatment in people, regardless of all the trendy drugs that are happening in Hollywood, are actually gastric bypass. And it's actually shown that gastric bypass actually alters the microbiome. So it's not just the physical restriction of calories, it's the alteration in the microbiome, and particularly with the impacts that has on appetite and stimulation of insulin secretion. So there are some other emerging connections that are, with the GI microbiome, and that's one of them is the gut-lung axis, and it's been shown that in a lot of true influenza type, or even with COVID, that there is a dysbiosis in the gut, and that there can also be associated, inflammatory changes associated with respiratory infections. There's also a lot of work being looked at or aimed at the gut-brain axis, and so this is a bidirectional communication. So where the brain, via the enteric autonomic nervous system, will actually alter the motility and the secretion and the mucous biofilm of the intestine, and then that in turn can alter the gut microbiota. The other way, the gut microbiota produces a whole bunch of neurotransmitters, metabolites, bile acids that affects tryptophan metabolism, and then can also stimulate the brain via the hypothalamic pituitary adrenal axis. So it's a very complex bidirectional communication. So what do we know about this gut-brain axis? So there's been a lot of studies to show that the GI microbiome is really important in rodent models for brain development. So again, that kind of brings us back to the parvovirus or antimicrobials in young puppies, learning and memory, and then also stress and anxiety. In people, there's a lot of work looking at hepatic encephalopathy and elderly dementia. We already manipulate the GI microbiome using lactulose when we treat hepatic encephalopathy. So, you know, we used to think we're probably underestimating how it works. And there is some suggestion that probiotic supplements may reduce stress to negative emotional stimuli. So the bacteria or the probiotics that have been most commonly assessed in rodent models include Bifidobacterium longum, and then Lactobacillus rhamnosus and Lactobacillus acidophilus. And all of those have shown in a lot of these rodent models that there is decreased stress or anxiety behavior, increased learning and memory, and altered expression of GABA in the brain as well. So the level of evidence in various diseases is pretty moderate in hepatic encephalopathy, depression, and Alzheimer's in people. And interestingly, with Alzheimer's, the GI microbiome changes actually precede any CNS changes as well. And in dogs, anxiety and epilepsy are emerging areas of interest, and the level of evidence is increasing to probably being fairly moderate now. So what we know in terms of manipulating the microbiome, we know that Bifidobacterium longum, which was isolated from some calm golden retrievers and has been released as a commercial probiotic, that's been shown to be effective at reducing anxiety, but also benefiting in kenneling stress as well. The other area of manipulation for stress and anxiety in dogs includes tryptophan supplementation, although that's probably a little bit less documented as being effective. We also know that medium chain triglyceride supplementation and ketogenic diets are quite effective for canine cognitive dysfunction and also reduce the incidence and severity of epilepsy. And this is associated with a switch in the microbiome as well as an alteration in the energy, you know, as it's transmitted, you know, energy as the brain uses it. So there's a lot of evidence to suggest that the microbiome impacts those particular conditions. There's also increasing interest in looking at the gut-kidney axis, because we know that when we have microbial end products that accumulate with uremia because they're not actually being filtered as effectively, it will increase intestinal permeability, which perpetuates a systemic inflammation. And then we will have an alteration in the way particular dietary substances are processed, and have alterations in particular bacterial families as well. And so there's been this sort of, there's a vicious cycle with chronic kidney disease and gut dysbiosis as well. And of course, we also know that dysbiosis occurs in both acute and chronic enteropathies. There's a bit of a lack of consensus in the studies that have been gone on about exactly what is the absolute change, and what is probably more likely is that there's a very highly individualized response in all sort of chronic conditions, and it is likely to be different between different breeds, different treatment groups, different samples, and different methodologies in analysis as well. And we also know that cats that have either acute or chronic diarrhea have quite an altered microbiome as well. just generally getting fecal samples regularly and as effectively in cats is certainly not as easy as it is in dogs. And we also know with cardiac disease that there is an alteration in the GI microbiome. So heart disease by definition will reduce sort of intestinal blood flow, and it can cause some edema that alters the GI bacteria. And then these intestinal microflora can actually produce oxidative metabolites that can potentially progress cardiac damage. And there's been a reasonable amount of work in people and in dogs that's beginning to explore whether that's an area that could, could be used therapeutically, to modify or slow the progression of cardiac disease. But one of the things I guess I'd like to say with a lot of these studies, or our biggest problem is we often have sort of like that chicken and the egg situation. So by the time we've got a clinical manifestation of a syndrome and we identify that there's a dysbiosis, it is a little bit hard to know whether the dysbiosis has proceeded and potentially caused the clinical disease, or whether the clinical disease has caused the dysbiosis because of systemic inflammation, or what other what other aspect is there. However, we know that most of the time it's relatively safe to modify the microbiome, and so in those particular conditions, there's a whole bunch of different ways that we can consider or think about ways to modify, and sometimes we do already. So what I'm going to talk about today is briefly how these work, not so much specifics, but we'll talk about prebiotics, antibiotics, probiotics, fecal microbial transplantation, and postbiotics. And when the term symbiotics is used, it's usually when there's a probiotic that's combined with a prebiotic. So I've kind of tried to use these graphics, and I'm sorry if it hasn't come out that well. But if you think of the intestinal tract as being this sort of river on the end, it's this wonderful ecosystem that has its own microorganisms and its own, own flora as well. When we're giving probiotics, we're basically dumping a whole bunch of fish into that river, and we're hoping that when we dump them in at one end, they are still alive and still effective by the time they come out at the other end of the river. So effectively that they don't cause harm, that they survive the gastric acidity, and that they are transported to the colon where they actually can exert their benefits. So with probiotics, they are usually only effective while the probiotics are being given, and the impacts and the changes in the microbiome are really transient. And so once the probiotic is stopped, then usually the impacts are stopped as well. So if we were talking about probiotic benefit in chronic enteropathy, so chronic diarrhea, which is, I guess, my major area of interest, there is some moderate evidence that they can reduce inflammatory markers and have some benefit in chronic diarrhea. And there's also some benefit in cats with acute diarrhea, more in reducing the days of diarrhea rather than changing the outcome. So if we have an animal with chronic diarrhea, I would probably not be recommending a probiotic as the sole treatment. Initially, diet with a prebiotic would probably be most effective, but they may be really useful for managing flare-ups, particularly in dogs that have food-responsive chronic enteropathy, or if there's a stress-induced diarrhea. So giving prior to being kennelled, or in a situation where there's been a recent shift and a potential stress exacerbation of GI disease. So when we're talking about prebiotics, so with prebiotics, the rationale behind this is that we are looking at feeding a selective substance that's going to increase the production or the rate of one particular bacterial species. So we're selectively breeding, I guess, or enhancing the breeding of a particular species of bacteria. So most prebiotics are fiber-based, although there's a lot of emerging interest in polyphenols and other products. And most prebiotics are going to be found in diets in the veterinary field. An emerging area that I think you'll find a lot of information about probably in the next decade are postbiotics. And so with postbiotics, this is where we're actually capturing the metabolites that are produced by the good bacteria, so effectively the fish poo, and then feeding it to the host with the hope that it will have the same benefits in the absence of the bacteria. And probably the areas that are looked at the most at the moment in people are using short-chain fatty acids like acetate and butyrate. And I think that's an exciting, potentially exciting area for us to, you know, think about in sort of future research. And so when we talk about fecal microbial transplantation, what we're effectively taking is we are taking a tank of fish, and that includes the water in which that fish swim in, their outputs, so their postbiotics, all the viruses that are swimming around with those bacteria and those fish, and then we're dumping it in the river. And we're dumping it sort of downstream rather than upstream. So FMTs are the treatment of choice for C. difficile associated diarrhea in people. And it is being increasingly used in veterinary practice. And it might be useful for a variety of reasons. It might be the bacteria that are present there, or it might be the bile acids and the you know, bacterial metabolites that are produced, or it might actually be the bacteriophages that are in these healthy donor samples. So when we do FMTs and when we are screening for donors, they're usually vet or nurse-owned dogs. So sometimes it can be challenging to find them that fulfill all this criteria. But we don't want them to be obese or to have skin disease because of the potential for them to transfer that phenotype. We obviously want them to be vaccinated or wormed with no history of GI disease and no recent administration of antibiotics or proton pump inhibitors. Additionally, it's better if they have no behavioural conditions because, again, the association between stress or anxiety and a dysbiosis. Generally, the way that I recommend screening them is a standard fecal examination, a clinical examination, and then a fecal dysbiosis index, just to make sure that you're not giving, you know, you're not using an animal that has a markedly altered microbiome. So the process is actually relatively simple if you are doing it in-house. That's where you collect you know, feces from donor dogs. You don't refrigerate the feces. It needs to be used within about 4 hours. You dilute the feces with saline and you blend it till it's liquid and remove the large particulate matters, because there's often quite a bit of bone and hair in the feces, by passing it through the sieve. It is then administered to the recipient. I generally fast them and, depending on their temperament, may consider to sedate or anaesthetize them. And so it is usually administered via like a soft catheter introduced rectally, probably up to the level of the transverse colon. And so this is how we perform FMTs, you know, in clinical practice, how I performed most of them. But there's been a lot of advances that suggest that, you know, there's a lot that we don't know. So, you know, is giving it into the colon, you know, in this way as effective, better, or the same as if it was introduced endoscopically into the ileum? There are some commercial providers that are developing FMTs in capsules so that they can be given orally with the capsule dissolving and not releasing the bacteria until the colon. And is that okay? These are the methodologies that are being studied in a lot of metabolic diseases, such as you know, such as obesity. If we lipophylize the bacteria, is that okay? Should we use more than one donor at a time? And that is something that I think is probably true. So generally, what we are doing at the moment is I usually use at least 2 donors, and I combine the FMT. And we don't really know how often we should administer them. And I guess there are some more guidelines that are becoming. So although it's being used extensively, the you know, the rationale to use them is not necessarily best practice yet. So what about antibiotics? I guess the way that I think about antibiotics is that it's like a really selective drag netting, and removing all bacteria from the river, regardless of whether whether they're good bacteria or not. So it's a very non-selective way of altering the microbiome. And what we know more recently is that particular antibiotics like metronidazole and tylosin, which are quite commonly used for managing diarrhea in dogs, can have really prolonged effects both on the microbiome, but then also on the metabolome, so also on the actual systemic health of the host, up to sort of 4 to 6 weeks later. And this again has been thought potentially to be associated with a decreased production of secondary bile acids. But it's highly variable and highly individual, and I guess there's a reason why I have the picture of those elastic bands on the right. If you think that each person's or dog's or cat's microbiome is an elastic band, you can stretch it, for a variety of times, and depending on the degree of the stretch or the frequency of the stretch, it's either going to bounce back to what it was very quickly, or it may remain out of shape and stretched and dysbiotic. And it's highly individual and highly unpredictable. So, in terms of using antibiotics in chronic diarrhea, there has been a recent study looking at dogs with chronic enteropathy or chronic diarrhea, and the dogs that were given metronidazole, although they seem to have a pretty decent response, and the owners reported that there was a reduction in diarrhea, when you looked at them over time, they actually had a poorer prognosis. So they had more relapses and ultimately a worse outcome than the typical food responsive or the steroid responsive forms of the disease. And this is regardless of the initial severity score. So even those that started off being quite mild. So currently, we really don't recommend the use of metronidazole or tylosin in chronic disease unless there's a specific pathogen that has been identified, or you're worried about overwhelming septicemia. And there are some particular breeds where it might be considered to be beneficial, but currently it's not the major recommendation. So that's kind of ended me in my 50-minute whirlwind tour of the GI microbiome, and I'm really happy to answer questions of any sort.
Caroline, that was fantastic. I hope nobody was eating as you were describing the process of creating your own fecal microbiome transfer process, or using a blender.
Yeah.
We have one question that's come in on the chat line, and it was actually in relation to that exact point. So when you were describing that FMT process, the question from Sarah, I think it means, was there no screening of the content of the fecal microbiome that you were transferring?
Yeah, so that's the Dysbiosis index.
Yeah.
So I think Dysbiosis index, a fecal examination and a clinical examination, and the Dysbiosis index is a nice thing to have, but I think if you eliminate all the other things, so you make sure they have no behavioural, systemic inflammatory disease. So if truly is a healthy, lean animal, do you run the risk of transferring a pathogen? Probably no. And I'd really question, if you've done your fecal PCR, probably no. So I think the risk is low of causing harm. I guess the only thing is that we don't know whether that individual would be, I don't know whether that individual would be of benefit. So but even if you had a Dysbiosis index, or even if you had sequencing or metagenomics of the donor, you still don't know that they're necessarily going to be the right match for the recipient. So sometimes it's a case of trial and error, and that's one of the reasons why often, more than one donor is used.
Thank you. Another question has come in asking if there's any FMTs currently being done for cats.
Yeah, there's been a little bit of research. To me, that just seems, it's just, for some reason, it seems more hard. Part of it is collecting fecal samples fresh from cats. So doing FMT in clinics, using it administered via an enema, is probably unrealistic, because you're just not going to get a fresh enough sample from your cats. And so healthy cats sometimes don't defecate, every day. So it'd be really challenging. But there are some studies that are looking at like processing and having it with FMT banks. And so they either lyophilize, so they basically freeze dry the feces, and then it's also stored and brought back when it's needed, and then using a capsule as well. So there is some emerging research in FMTs in cats.
Okay, thank you. There's another one that's just popped in. It's quite a long one, so I'll read it out to you. So AR has a patient who's had more than 50% of his bowel resected as a puppy, and now has a problem with body condition, and has had that since. This patient is on a myriad of medications and prebiotics, which seem to all help, including intermittent short courses of metronidazole and occasionally enrofloxacin. Would this patient benefit from obtaining Dysbiosis index to obtain changes to this patient's treatment plan?
Well, possibly. So that's like short bowel syndrome, I guess. And there's a whole bunch of things, that I would look at. I still would be really reluctant about using metronidazole, and I particularly wouldn't want to use enrofloxacin. But I say that from a theoretical point of view. I guess that.
Sorry, Caroline, that was my fault. It was tyrosine and metronidazole.
Okay, that's good. So often with short bowel syndrome, it's a question of trial and error. But the first thing is to try the different diets. And so I would start with like a highly digestible, low residue diet, or potentially add in, if that doesn't work, then add in a hydrolyzed diet, and then go for quite an elemental or a modified fat diet. So either using a low fat diet or a diet that's supplemented with medium chain triglycerides, just because they're not absorbed in the same way through the GI tract. So it's likely to cause to be slightly better. And you probably need twice the resting energy requirement calorically for dogs with short bowel syndrome as compared to those that have a normal intestinal tract. If there has been ileal resection, I would be worried about whether there's a bile acid deficiency. I would look at, obviously giving any highly digestible, low residue diet. If that doesn't work, I'd try a hydrolyzed diet. Then I would make sure, that that all your micronutrients have been addressed as well. So cobalamin, folate, vitamin D. So you can measure all of those and supplement all of those. And then if you're still not getting your weight up, then I would try bile acid sequestrant.
Great. AR has come back with one last comment. He would like you to provide some examples of diets that you just mentioned. I can do it from Purina's perspective, but it's
so like every company has one. So yeah. Purina's would be HA, so hydrolyzed antigen diet. The Royal Canin digestive low fat would be another option in terms of like an ultra fat restricted diet. And then a hydrolyzed diet could be Royal Canin hypoallergenic, or Hill ZD. Yeah,
that's great. And just while I can, I will say that our EN, our gastrointestinal diet, also uses those medium chain triglycerides that you mentioned earlier too.
Yeah, I don't think the fat's quite that high. It's kind of a moderate fat content though, isn't it?
We've just launched EN low fat in Australia.
Okay, there you go. There you go.
Anyway, another question. When would you consider an FMT trial in patients with chronic enteropathy?
So once they've failed their diet trial.
Yeah.
And it has to be like a real diet trial. And, I usually have to try 2 or 3 different diets.
Julianne has put a question, and I'm not quite sure. I'll read it out to you. Okay, I think it's based on a previous recommendation. So previously, antibiotic trials were recommended before biopsy and immunosuppression. Based on what you've shared tonight, re tyrosine and metronidazole, has this recommendation changed?
Absolutely. Yeah. Don't use them.
Along with enrofloxacin.
Well, enrofloxacin, like I just want to put the little caveat out there, unless it's a boxer or a bulldog, French bulldog that has granulomatous colitis, in which case, yeah, enrofloxacin is pretty effective. Yeah.
Okay. I can answer this one. Is the ProPlan Elemental diet becoming available in Australia soon? No plans at the moment, but I would say watch this space on that one. We'd love to get it here.
Yeah, and that's another type of diet that you can try for short bowel syndrome as well if you need to. Yeah.
Another cat question. Cats with IBD that mainly present for chronic vomiting, would FMT be an option in those cases?
Theoretically, I just don't know that we know enough yet, to be honest. And I think with chronic vomiting, I'm always concerned that we're missing a small cell lymphoma. So with cats, I'm quicker to aspirate and quicker to biopsy, and I quite often skip from the diet straight to the anti-inflammatories or immunosuppressive modification. Yeah. But if you speak to 5 different gastroenterologists, you'll probably get 5 different answers for that.
Let's not even talk about speaking with behaviorists. but actually, you raised some really interesting questions about that gut-brain axis. But yeah, another question has come in. What bile acid sequestrants can be used in dogs?
So I'm gonna butcher this pronunciation. So I think it's called cholecysteiramine. Yeah. There was a recent paper in JVIM that talked about bile acid diarrhea that has the dose rates and everything in it, and it's open access if anyone's interested in looking at that.
Okay, great. I'm curious. I think there's any more questions that have come in. If I haven't answered your question and you've typed one in the chat, please retype it so I can call it out to Caroline. While I'm waiting for that, I'm just curious about. You were talking about the impact of when we were talking about the gut-brain axis and the changes in epilepsy associated with supplementing with medium-chain triglycerides. And we're well aware of the ability of the gut-brain axis to absorb those ketone bodies produced by the MCTs as an alternative energy source to glucose in these idiopathic epileptic dogs, but you alluded to a change in microbiome, and I'm curious as to what that change would be and the impact that might also have.
Yeah, and I think the problem is we don't really know, because the studies they've basically shown that there is a shift in the microbiome, but what we don't know is whether there's a functional impact of that shift in the microbiome. Certainly you're right. It's the ketogenic aspect that has been probably the most documented, but then serendipitously there's been a change in the microbiome. Now, whether that's because the gut-brain axis is healthier because the brain is feeling better, we don't know, and that's kind of where it comes to this. Is it a cause or effect or an amplification of the disease? And so there's a lot of questions that we don't know. But yes, there's been some recent, very, very recent, like in the last 6 months, papers that have shown that medium-chain triglyceride diets in epileptic and dogs have an altered microbiome.
Okay, so loads more research in this area.
Yeah.
Well, Caroline, I think we're just about up for time. Really want to thank you for sharing all of that information. I think it was super informative. I also loved your graphics, by the way, from the obese mice all the way through to the rivers and the fish.
Yeah. Thank you.
No, I really, really enjoyed it, and from the comments coming through in the chat line, I think many people who registered enjoyed it too. So hopefully we can maybe ask you along again another time to do another webinar for us.
Yeah, that'd be great. All right.
Thanks, Caroline. For everyone else, we will be sharing this on demand, so it has been recorded, and we will send you a link to the recording in the next week or two, which will be available on YouTube. So thank you so much for your attention, for joining us tonight, and I hope you can take some of what you've heard into your own practice going forward.